Hi, good morning to everyone listening from Europe, the Middle East, and Africa, and to everyone tuning in from Asia, good evening. If there's anyone joining us from the Americas, thank you very much for joining us at this ungodly hour for you. My name is Gabriel, I'm from Imperial College, and I'll be hosting this webinar on behalf of the Early Career Members Network. So I'd like to welcome everyone to today's live webinar, brought to you by the Early Career Members Network Committee of the Metabolomics Society. This is the third in our webinar series, and we'd like to thank everyone for their support and invite everyone to keep on tuning in to the rest of our webinars. This session, as you may already know, is free for all to attend and listen in, and the recording of the webinar will be made available afterwards for all registered members of the Metabolomics Society. If you haven't already, please connect with the Metabolomics Society online on Facebook, Twitter, LinkedIn, with all the information shown on screen. We're also very keen on feedback and suggestions from all the Early Career Members So if you have anything in mind, please send us an email at info.emn@metabolomicsociety.org. In addition, just a quick reminder that Metabolomics 2015, the International Conference of the Metabolomics Society, is coming up at the end of June of this year, and the EMN Committee will be hosting a welcome event and reception on the 28th of June, that's Sunday, from 4 to 6 p.m., and we will also be hosting 4 EMN workshops on Monday, the 29th of June, throughout the day. So we'll be giving more details of these workshops later to remind you after the webinar. So today's speaker is Professor Bas Teusink, Professor in Systems Bioinformatics at VU University Amsterdam. His work combines theory, modeling, and quantitative experimentation to understand the design principles of metabolic networks and their regulation. So this knowledge of metabolism is then applied to fundamental as well as applied problems in cell biology, biotechnology, and medicine. There will be time after the webinar for questions, so if you have any, please type it in into the webinar console. There should be an option on your webinar console that says questions. So you can type it in, and after the webinar, we'll be forwarding these messages on for Bas to answer live. So with that, I will pass you on to our webinar speaker, Professor Bas Teusink.
Okay, thank you very much, Gabriel, and welcome to this seminar. It's my first webinar, I should say. Thanks for listening. Let me see if this works. Yes, so the talk I want to give is about the use of models as a guide and a natural integrator of data, and in particular metabolomics data, as I will show. Now, a lot of omics data is being generated out of ignorance, which basically means we don't really know all the components and their role they play. And as Douglas Carroll and Stephen Oliver pointed out some time ago, this is just one part of the science cycle that we have, which is both inductive and deductive. And so the message I have is that instead of just only generating data, metabolomics data, we can actually use models, and that's the message of this, models, mechanistic models, as a natural way to integrate data. And these models are available, especially for metabolism. And the most logical models, and the ones that are most popular at the moment, are so-called genome scale metabolic models. As you can see here, this is an example from an organism that I study. And these are just metabolic networks that are derived from the genome. So we start with the genome, and using bioinformatics tools, we can then fish out these genes that encode for proteins that are enzymes. And then using metabolic databases, such as KEGG, you can figure out what the reactions are that these proteins catalyze. And in this way, you can get a network, a specific network, metabolic network of your sequenced organism. Now, making this model is laborious, but it's not that you at the end have a model and the model is really a bottleneck. No, you can use this model along the line to try to understand more and more of your organism. And along the line, you will get new insights that you can actually use already through applications. So I would see a model as a sort of an evolvable knowledge base that you can use for all sorts of applications. Now the nice thing about a model is that you can actually compute with it, and so there's many applications of these models, and for these genome scale models it's all centered around what we call mass balancing, and it's basically just trying to account for what comes in and what comes out of a network. So in most cases this would be nutrients coming into the network, as you can see here, and then as an output you'll have new cells, also biomass, products you might be interested in, and in many cases also byproducts that you might not be interested in. And so what we basically do is we keep track of all these metabolic fluxes in the network. And this has been very successful for metabolic engineering, trying to figure out where to interact with this network, knock out genes or add new genes to see if you can get more product. Also for drug targets, this has been used. It's very useful to get basic physiological knowledge of your organism that you've just sequenced. And what I will show today, also in the case of medium optimization. So what on the nutrient side you can change to improve your process. They've been very useful, and I will give a use case of that. So if you talk with companies, then the sort of the traditional way to improve processes through the medium is by what they call design of experiment, which is basically probing in the dark. You have a medium and you systematically start to vary different components in the medium and possibly in different combinations to see which one will improve your product. Now, in the case that we have, which is a human pathogen, we had 27 compounds in the medium, and so you can imagine that you need many combinations of nutrients, many cultivations to get an idea of what would be the best combination. And this is not a very sustainable way of doing things if you have complex media. And what you basically do not use in such a blind approach is the knowledge that we have of metabolism and the stoichiometry of reactions. And so in a metabolic modeling approach, it would be more rational. You can actually use this knowledge, and I will show that with a very limited number of cultures, you can actually get much better predictions. Now, before we can do that, we need to make a model. And so the way we do this is we start with the genome. And one way to get quickly to a draft model is to actually see, which genes in this genome are ortholog to the genes in already made models. And we have many of these models already available. And so we copy reactions from these models and we get a draft model. And then we can start to model and play with this to assess the model performance, do model simulations, and then compare these model simulations with literature and experimental data. And then we start an iterative, very powerful cycle of modeling, experiment, modeling, experiment. Okay. Now, the reference fermentations that we did in this case were just two batch fermentations over a couple of days, everything controlled, as you can see here, oxygen, pH, and these sort of things. And what we did is an extensive characterization of these fermentations using metabolomics. And so in this case, the exometabolomics, so what's in the supernatant. Here you see an example of a number of measurements that we did. So you see here the increase in biomass in the two fermentations, and you see that some things are being produced, like ammonia here, and some things are being consumed. And so we did a full time course of biomass and 39 extracellular metabolites using all sorts of metabolomics measurements. So the other thing that we need is the biomass, because this is what, in this case, was the main product of the fermentation. It's the biomass. This was for vaccine production, and they needed the biomass in the end to make the vaccine. So they were interested in biomass. And so we did an extensive characterization also of the biomass. So looking at the amino acid composition, as you can see here, the lipid composition, lipidomics and literature, cofactors, RNA, DNA, all these components that you need to make a new cell. And also during the fermentation, we measured total protein, total RNA, and lipid content to see if there are major differences during the fermentation. And as you can see, there's not many differences. Perhaps on the RNA level there is, but for most components, these were pretty constant in this case. Okay, now having these data, you can actually start to use a model to analyze. And the first problem that we found in these fermentations is an imbalance in nitrogen. These pathogens, they grow on amino acids, and of course they contain nitrogen, and they contain more nitrogen than the biomass does. So we're having a massive imbalance in nitrogen. So if we try to close the balance, we found that about 37% was excreted as ammonia, and then because we had the biomass, we could figure out how much nitrogen is in the biomass, which was another 30% or so in protein and in nucleotides, RNA, DNA. And this is where it ended, and we were pretty much clueless what else. And here's where the model comes in. So now you can ask the model, if you pour in so many amino acids, and this is coming out, what else can come out of these models, and what would be actually the best way to get rid of your nitrogen? And the model suggested arginine. And arginine contains many nitrogens, so this will be the optimal way to get rid of the nitrogen. And indeed we found, with targeted metabolomics, we found that arginine is being secreted, but not so much. And so here we were left in the dark. And as I will show you, we used LCMS to try to identify more of these compounds. And in the end, we could, at this moment, we have about 10% of the rest of the nitrogen closed. Now, the way we did this, is we used an LCMS-MS detection method. This is not really a state-of-the-art machine for comprehensive metabolomics. But we used the machine together with modeling, and I'll explain how this works. So the first thing we did is we did a total mass screen of the supernatant, and then we could find 88 and 20 signals in positive and negative ion mode that were proportional to dilution, so giving us an idea these were real signals in supernatants. And then we used a metabolome database to find out, based on the MZ score, which potential metabolites these could be. And we get many hits, as you can see here, 12,000 and more than 1,000. And this is, of course, then a nightmare. But because we have the model, we know from the model what compounds can be secreted based on the knowledge we have. So if we filter against our model, we actually came up with 65 candidate compounds that matched the supernatant and the model. And then for these 65, we tried to do a more targeted approach, to do selective reaction monitoring. We could get our hands off, in the end, 32 compounds where we could get these transitions. And then we could use that to do targeted LCMS back on these supernatants. And in this way, we identified 10 compounds that were all synthesized in medium. And then we could do quantification by standard addition. And these compounds, you can see here, most of them were actually very unsuspected, and they are nucleotides, as you can see here. And if we quantify these, if we try to quantify these, we could see that in particular adenine would accumulate up to about 10 millimolar, which is really a high concentration for such a valuable nucleotide. And it's clearly from the model not the most optimal way to get rid of nitrogen. And so we suspect that this actually has a functional role in the host pathogen interaction. And if you then dig into the literature, you can actually find that indeed adenine has been described as an immunosuppressive agent. So this is an illustration how a combination of first untargeted metabolomics and the model can actually lead to this type of discoveries. Now, this is not medium improvement. So how can you use these type of models for medium improvement and get better productivity? Now, if you have exometabolome data, you have a biomass composition, and you have a model, you can start to do an optimization. In this case, maximizing the amount of biomass. And now we can use the measured nutrient uptakes and the measured byproducts as constraints in the optimization, and then ask the model, okay, what's the maximum amount of biomass, and how are these constraints limiting that amount of biomass? And this is called the reduced cost analysis, where you ask which nutrients, which constraints, I should say, and limit the output, in this case, growth, biomass production. And then you get a list, if you play with this, you get a list of compounds that potentially can limit biomass, and therefore if you would increase it, you might get more biomass. This is, and depending on how big the effect is and how far away you are from the constraints, you could prioritize which compounds seem to be most likely, and so you can come up with the recommendation list for the experimentalist then to test. And that's what we did. So we have here some suggested changes, and here you can see a number of experiments. These are quite a few experiments, much less than you would need for a design of experiment. And you can see that we can get, in some cases, a twofold increase in biomass, which fitted quite nicely with our predictions. I think on average we had about a 70% success rate with these type of predictions. So this shows you how you can use such a model together with some data to quickly get to the right experiment. So you eliminate a lot of experiments that based on the model would be nonsense to do. But you can go a step further. So you can say, okay, I have my model now, and I don't necessarily need to be constrained to the medium that in the past people have designed. In many cases based on trial and error or on history. You can really start to think out of the box. And so one thing that we did is we developed an algorithm, it's called EMF, for enumeration of minimal active fluxes, where we ask, okay, if we have a set of exchange reactions, so these are all the nutrients that can potentially be taken up by an organism, what would be the minimal number of fluxes that you would need to get growth? That will be the objective function in this case. So basically what we need to do is we need to set, we have a model, we set some condition-specific constraints and a list of reactions to be minimized. Then we get the minimum number of active fluxes, and then we can use a technique is called flux variability analysis. We can actually try to vary different fluxes within this minimum number that we have to see if there are groups of alternative active fluxes. So different nitrogen sources, different carbon sources that can support growth. And you can also find some nutrients that you always need. There's no alternative, and these will be really the auxotrophies of this organism. They are needed to grow. And here's an illustration of that. So you can set constraints and then do the minimization. So you can maybe say, okay, I want to have the absolute minimal medium, the minimum amount of compounds. And there we were actually surprised because this pathogen that I'm talking about, people thought it's a fastidious organism and it needs many amino acids to grow on, but we actually found based on the model that it only needs cysteine as the only carbon, sulfur and nitrogen source. It's a big surprise, and this is actually what we validated experimentally. Actually all the examples you see here are validated experimentally. This is a really minimal medium, but you can also say, suppose we have only amino acids available, so you set different constraints, you can do the same thing, and then you can see that there are sulfur sources and there are now a combination of carbon and nitrogen sources because all amino acids have carbon and nitrogen. And you can see which of these amino acids support growth. And basically most amino acids do. You can go one step further and say, no, I don't want amino acids, I want a minimal media. So amino acids are excluded. And now what potential other combinations of nutrients can support growth? And here you see that this organism can grow with, for instance, ammonia and different types of carbons. But there's different combinations that are possible here. Another interesting thing that we found is that this organism can grow on an inorganic form of sulfate. It doesn't necessarily need a cysteine or methionine or glutathione to grow on. And this was key to the improvement of the vaccine production, as I will show in a second. So based on this enumeration of all the potential possible media that this bug can grow on, we came up with more than 4,000 different media, which is of course a lot to test experimentally. So you can then set additional constraints or wishes that you want. For instance, substrate availability or the price, solubility, and these sort of things. And with that, and also knowledge that the company has on what are better compounds to work with than others. And then in this case, we came up with 8 different chemically defined media compositions that were patented. And what we saw is that, as I said, we could find this alternative inorganic sulfur source. And that was interesting because they knew that sulfate is a inhibitor of the toxin that is being produced with the biomass. That's actually an important ingredient of the vaccine. And so sulfate is a natural byproduct of amino acids, sulfur-containing amino acids. And this sulfate builds up at the end of the fermentation. Now, if you can change that into an inorganic sulfur phosphate, which is now as a substrate, then this substrate will decrease in the fermentation. And you would expect at the end of the fermentation to have very little of this sulfur compound present. And indeed, that's what we found. If we do that, we got a 4.4 increase in productivity. So to summarize the approach that we take here and how we got to this massive improvement in the vaccine production is we have experimental data, we have a draft media, we have genomics, bioinformatics, that makes models. These data and model sets constraints, and with these constraints we can come up with media compositions that would work best. And so, as I said, in the end, this is the massive improvement that we got from 15.2 to 36.7 improvement in biomass. And remember, this was with doing 2 fermentations, a few specific model-driven growth experiments to validate some of the predictions, and that's it. So if you compare that again with the design of experiment, which is at the moment, I think, the traditional approach in most companies, you would need to do massive amounts of cultivations. And this is a much more efficient way to do things. And, as I said, you also learn much more. So yeah, that was the 2.4-fold. So you learn much more. During the reconstruction, we found all sorts of interesting new things. So we found adenine secretion, so that points to a host pathogen interaction that might be druggable at some point. We found many more things like the TCA cycle, for instance, turns out to be complete in this organism. And so there's many things that you can, along the process, during the model construction, you can learn and you can actually use to your advantage. Okay, I hope I brought this message. So the people involved, this is my group. My group is doing systems biology and bioinformatics. We had great help from the analytical chemistry group here at the VU. Who did the metabolomics analyses, and we worked together with GlaxoSmithKline, who also did a lot of experiments and guided us through the biological questions. And with that, I'm done, and I think I'm reasonably in time. Okay, thank you very much.
All right, great. Many thanks to Bas. So now we have time to accept questions. If there are any questions from the audience, there should be a panel on your control panel that says questions. Just type in your questions and we'll get to those in due course. So yeah, we'll give you a couple minutes to type those questions in, and we'll pass it on to Bas. But yeah, I guess I'll kick off the first question while we wait for more to come in. My question is, when you're building the models from information from literature, how difficult is it to adapt to your own purposes? Because I've heard of people who've struggled to make published models operate in the same way that they've been described in the publication.
These seems like two different questions. So how easy is it to incorporate? It depends. Ideally, you would really have the access to experimental data. So as I said, you would need to have an idea of the appearance and disappearance of metabolites in the medium, so that you can set these as constraints and you have some data to validate your model. And also on the biomass, that would be useful, although in most cases biomass compositions, the bulk will probably not be very different between different organisms. So for instance, protein is always about 50% of biomass, so it's a bulk, really bulk. The differences is mainly in most cases in the cell wall or in the lipid composition. So that's where specific data would be very useful. The second question you had, you said people have problems publishing these models or reproducing these models. Can you
reproducing mostly, is what I've generally heard.
Yeah, that's a big issue in the field, and people work hard on this, and this has to do with lack of standards, which I think is a problem in all of biology. So people are working hard on developing standards for encoding these models, and also to use the right identifiers for instance, the metabolites. So I think Chebai identifiers is currently the way forward to make sure that all these models that are being published have these type of identifiers. So, and you can also link experimental data to the relevant metabolites in these models. So standards is really the key. We have developed also a standard. SBML is maybe something that some of the audience is aware of. It's a systems biology markup language. It's a standard, an XML-based standard for exchange of models, and this is now also available for these genome scale type of models. It would allow exchange of models across different modeling platforms. Does that answer your question?
Yeah, it does. I've got another question from Alfredo Ibanez. The question is, how do you build a model when there isn't any genetic data available?
If there's no genetic data available, you're in trouble. But I guess these days, getting the genetic data, the sequencing is not the bottleneck, I would say. And actually, one of the ways where the field is moving is to look at metagenomics data and then try to build metabolic networks of communities. That's the new frontier, I think, in this. But you need the you need the genetic information. You need the sequence.
Okay. So are there any more questions from the rest of our audience? All right. Well, we can give them a few more minutes to send them in and then come back to it if any come in during that period. But yeah, again, thank you, Bas, for delivering this webinar today. Again, to remind everyone that we have our EMN Welcome and Reception at Metabolomics 2015 on the Sunday, 20th of June, 4 to 6 p.m. And we will be holding 4 EMN workshops on the Monday, the 29th of June. And just to give a few more details on that, just a reminder that early registration closes basically tomorrow, but regular registration closes on the 30th of May, and you can register late all the way until the start of the conference. Our workshops that we'll be holding on the Monday, first will be on big data, which we will be having Professor Douglas Kell from the University of Manchester and Dr. Rob Davidson from GigaScience. We will also be holding another workshop on fluxomics, pathway analysis, and metabolic engineering with Professor James Yao from the University of California, Los Angeles, from Professor Akihiko Kondo from the Riken Institute for Sustainable Resource Science, and Professor Uesauer from ETH Zurich. We will also be holding a workshop after that on careers in metabolomics. We'll be having speakers from academia, industry, and government with a full lineup to be confirmed afterwards, as well as a session on research integrity with Professor Roy Goodacre from the University of Manchester and Dr. Philippe Roccaserra from the University of Oxford. Again, a reminder, if you haven't already, please connect with us online on Facebook, Twitter, LinkedIn, or by email on the details sent below. So going back to questions, we've got a question from Sastia Putri, and the question is to Bas. What advice would you give any early career members who are just starting research on metabolomics and systems biology? And what would you say is the most important point to consider when designing experiments similar to the ones that you would pursue?
I would advise young career students to have sufficient knowledge on these type of modeling developments that go very rapidly now and statistics, so that you can effectively communicate with modelers if you do not want to do it yourself, and to establish an environment where the experimental biologists and the modelers really communicate and work together. So this was a success story only because at GlaxoSmithKline, where we collaborated with, they were receptive to the models and also to the modeling suggestions. And this was because in the company there was one person, Philippe Cauvin, who actually understood what we were doing. So if you want to use this effectively, and I think this is really the future, you should get enough knowledge on these methods to be able to communicate. That's the minimum that you would need. And it would make really your research much more powerful, as I illustrated.
All right, I've got another question as well from Sean O'Callaghan. What would you comment? How would you perceive the other simpler and more limited modeling approaches presented in the literature? Perhaps like kinetic flux profiling and maybe flux balance analysis. Would you say that this is worth doing, or should the community invest more in more detailed modeling along the lines as you have spoken about in the webinar?
I think sort of my mantra is that models should be useful, and so a model is useful if it's attached to a question. So it's very difficult to, in general, say what's the best modeling type. So in this case, we were really interested in the medium optimization, making biomass. So there, the global approach was the appropriate approach. If you're more interested in dynamics, and then you would need to have a kinetic model where you can really also look at the effect of feedbacks or other dynamic behaviors. So it really depends on the research question you have, and obviously also on the sort of data that you have, what would be the best modeling approach. There's no general answer to that.
Okay. We have another question from Matthias Steiger. On the experimental data that you had shown in the study, you showed that, for example, cysteine had a positive impact during media optimization when you added it, but you also found some other substances which didn't show any change. So some of the items in the model aren't really predicted all that correctly. How do you integrate this data into the model, back into the model to improve it further?
Yeah, that's a difficult question because, well, it's not a difficult question, it's a very good question. So I see models more as a tool to get hypotheses or to exclude experiments that would probably not be very useful to do, rather than being spot on predictive. There's so many things that we ignore. In these genome scale models, we only look at stoichiometry. So A goes to B goes to C. There's no kinetics in there. So if you now start to change the concentration in the medium, then it also depends, of course, on the kinetics, whether this would have a flux effect or not. And this is something we basically do not take into account in these models. So you have to accept that your success rate will not be 100%, But it basically, as I showed, excludes you from doing a lot of stupid experiments. And it's really also the exploratory nature of models. It sharpens your intuition. You can start to play and you build up in your head sort of an idea, okay, so cysteine seems to be important, the other one not. It's more this playing and getting this intuition, I think, that is the best use of these models, rather than try to really be spot on predictive. We're not there yet for that.
All right. And we've got one more follow-up from Alfredo Ibanez. So in the case of, say with a cysteine result, would you say that generating a genetic mutant of the organism would be required to validate the result of the model? Or is that not the approach you would take?
Well, it's a way to validate models. Actually, one of the ways that people try to validate these models is by predicting the phenotypes of knockouts. In this particular, and again, this goes back to the usefulness of the models. We use the model to predict future experiments. Some of them work, so the company is happy, right? So you can go on and try to validate. Now, knocking out, well, what you could do indeed is say, okay, so it grows on cysteine, and I predict a specific pathway, and I care about this pathway, and I want to validate that indeed it's via that pathway that cysteine is assimilated. You could, of course, make knockouts to see if it then cannot grow on cysteine anymore. Or you could maybe do a flux analysis to show that the flux goes through that pathway. So it really, again, depends on the question and the purpose of these models, whether you want to dig into that. But indeed, knocking out genes is a valid approach to see if the network that you've built in your model is the right network.
All right, we'll take one last question, and that's from Thomas Lawson. What's your experience of some of the off-the-shelf tools to generate models? Like the example listed was pathway tools from Biosite. And I guess what would your other recommendations be, and what is it that you use in your own day-to-day modeling work?
So there's a number of tools that would generate draft models. I would call them draft models. It's a good first start. In our experience, maybe 80% or so of the reactions, maybe 90% will be okay. But you do need to have somebody with an understanding of the organism or physiology of the organism to decide on the right 10% that's left. So some of the automatic model reconstruction tools, they will just fix it for you. So Model Seed, for instance, is a tool where you will get in the end a model that can produce biomass. But if you then look at the fixes that these automatic reconstructions make, then in many cases these are mistakes because they were copied from E. coli, and this is not E. coli but another bug. There's maybe transporters they add that this organism doesn't have. So the last 10% you need some expert information or manual creation in order to get it right. But again, also here it depends on the research question. If you want to compare 1,000 different metabolic networks, then this is manual creation is not an option, and then you have to accept that 10% of your reactions is wrong.
All right.
I
guess the other question would be what is it that you use for your own group's work? What software is it?
Okay, so we have actually, I'm fortunate to have a programmer in my group who is developing Python-based software. It's open source. It's available on the web. One of them is called FAME. Another is called Pisces. This is sort of homemade software for the reconstruction and also the modeling of these. So part of it is web-based. There's also tools on command line. So this is what we use. Most other people will probably use Cobra, but this is MATLAB based. And so for instance, for companies, this is not really an option. So we have open source tools available.
All right, great. So I guess that's all the questions we have for today. If anyone else has any more questions to put forward, just send us an email and we'll try to connect you with that and have answers to any further questions, maybe up with the recording. So again, I'd like to thank Bas for speaking at our webinar today, and thank you everyone for attending. We will be having another webinar in a few weeks' time, and more details of that will be sent once we have confirmed details for our speaker dates and times for everyone to find out. So thank you for attending, and hope you have a good rest of the day today. Thank you.